So for this question, number 4, erythropoietin, which is known as EPO, is a large glycoprotein synthesized by specialized cells in the kidney. So these are all important information. Specialized cells in the kidney will produce the EPO. Okay, so these cells are very sensitive to changes in oxygen concentration in the blood passing through the kidney, and it responds to low oxygen concentration by increasing the synthesis of EPO. That means to say that as the blood passes by the cell in the kidney, when the oxygen concentration is low, it's going to activate or stimulate those cells to produce more EPO. Okay, so what does EPO do? Yeah, I'll let Niran in, okay? So what does EPO do? EPO acts at the surface of a particular target cell, such as cells in the bone marrow. So a lot of information. So bone marrow is the target cell. Sorry, not bone marrow. Cells in the bone marrow are the target cells. These bone marrow cells are stimulated to produce red blood cells. So basically, if you want to put the passage into a summary, then it must be low oxygen concentration. So that will lead to the target cell to secrete more EPO, and this EPO target cell is the bone marrow cells, which bone marrow cells will actually produce more red blood cells. So this is summarizing the passage. Right? So this is exactly what happened. So when you can see this clearly, then answering the question is very easy. So it's good that you write this down so that as you continue to answer A part 1, part B, part C is related to this passage, right? Because A, B, C are all under this passage, right? You can see. So the information is very important. A part 1, a low oxygen concentration also leads to an increase in the quantity of mRNA in the specialized cells in the kidney. That means to say that they say that this specialized cell have got more mRNA being synthesized. The question suggests and explain why there is this increase in quantity of mRNA. So from the passage, you already know, whenever there's low in oxygen concentration, target cell will have to secrete more EPO. So in order to secrete EPO, it must first synthesize EPO, right? In order to synthesize EPO, then it's coded by the gene. From the gene, the code for the EPO, gene for EPO. Then you have to undergo transcription, isn't it? Followed by translation. So translation will give us the EPO. Let's say if this is a protein. So transcription will give us the mRNA. Right? So therefore, why there's an increase in mRNA? Then you look at the mark schema. So because a gene that codes for the protein, or actually the EPO, is being transcribed or being switched on, or there's an increase in gene expression of this particular gene. Either you must say it codes for protein or it codes for EPO. Okay, so gene coding for protein or EPO is 1 mark. Is being transcribed or being switched on or being expressed 1 mark. Therefore, and then mRNA for EPO synthesis is produced, or mRNA are produced to be translated into EPO. So that's the answer. The gene, the transcription, what is the use of the mRNA for? For EPO synthesis. Okay. Right, next question. This question is very, very easy. So this is a diagram of an mRNA. So this is an mRNA. Okay, so therefore you can see U, the base U here rather than T. So question asks a student, state the base sequences of the tRNA anticodon complementary to codon 1 and DNA sequence which code for codon 3. So codon 1, if this mRNA, tRNA must be anticodon must be complementary, right? So it's GGC, so very easy. First answer is GGC. Second one, what about the DNA sequence which code for codon 3? So it must be complementary, right? So complementary to codon 3 is actually CTA. So answer is GGC and CTA. Okay. Next, the 3 codons in figure 4.1 are near the start. This is important, huh? Near the start of the sequence coding for EPO. So it's not the start, but it's near to the start. So which means to say, in this diagram, codon number 1 is not the start codon. Okay, it's not the beginning. They say that this 3 codon here, this 3 codon here, okay, is near to the start. So don't know where is the start. So it's near to the start. They are not the start. Okay. Explain the consequence of a mutation which deletes the U from codon 2. So 4 marks. So what is the consequence of deleting the U from codon 2? So we are talking about EPO synthesis, right? Okay. So this is being deleted in codon 2. So when you know that when this is being deleted, that means to say that now the second codon will be occupied by AAG. So this is the second codon already. Then the third codon becomes AU something. So you can see that first thing is the second codon will be coding for a different amino acid. Because from UAA becomes AAG. So it's a different amino acid. Then followed by subsequent codons. That means 2 onwards, codon 2 onwards. Subsequent codon is actually codon 3, yeah, because I mentioned 2 already. So codon 3 onwards. Also code for different amino acid. Code for different amino acid. Okay. So this is the marking point. So this is the first one, this is the second one. Second one. Okay. Then third one, there's a frame shift mutation. So you can see that the frame, right, the UAA now becomes AAG. So the shift in the bases of one frame to the next frame. So there's a frame shift. So there's a frame shift mutation. Okay. So another thing to take note is the original UAA. Can you tell me what are the stop codon? Gia, what are the stop codon? Gia, what are the stop codon? You need to know. You don't know, Miss, go and memorize it. Gia, what are the stop codon? Okay, open to the floor. Gia cannot answer. Open to the floor. Who knows what are the stop codon? Akila? Give me the stop codon. UAG, yes. Got 3. UAA, correct. One more. UGA. Okay, so the stop codon are UAA, UAG, and UGA. What is the start codon? Let's start codon, AUG, right? Which are code for methionine, isn't it? Okay, so can you see that this example here, right? The UAA is a stop codon. UAA is a stop codon. So that means to say, based on the question, right, this mRNA, right, the transcription will stop here, no? So there's transcribed first amino acid, second amino acid, maybe third, the fourth, after that they stop. This is the question, huh? So after mutation, what happened? So that means to say that the stop codon, this stop codon is already removed already. So chain termination will be later in the mRNA compared to original is earlier. Right now, after mutation is later. So the chain termination should be later, which means to say the polypeptide chain coded will be longer already compared to the original without mutation. You see that? Okay. So because of the different amino acid sequences, right? So what happened is, of course, you will change in primary protein primary structure, including secondary structure, tertiary structure will change. So when the structure changes, that means the protein may not be functional. That means cannot function as usual because the shape has changed. Can you see that? So basically it's talking about change in base sequence, in DNA base sequence changes. Leads to mRNA base sequence changes, leads to amino acid sequence changes, so leads to protein structure. What's my next one? Protein structure changes. So we talk about primary, secondary, tertiary structure changes, and finally protein function changes. So basically these are the marking points. These are the points. So let's look at the mark scheme. Exactly what points they give mark. Huh? So this is the mark scheme. So the amino acid coded by the second codon will change. Therefore, amino acid coded by every subsequent codon from number 2, that means number 3 onwards, will also change. So there's one mark given if you say amino acid or the protein sequence or amino acid sequence before codon 1 until codon 1 will not be affected, which is true, isn't it? So those amino acid up to codon number 1 will not be affected. Okay. Idea of further chain termination. That's why the original mark scheme is wrong. Premature chain termination. So student who do past year and memorize past year, you write this answer is wrong lah, because past year mark scheme is wrong. There's no premature chain termination. It's actually later. Okay, because the chain will be longer. Idea of change in primary, secondary, tertiary structure of protein. I cannot write quaternary because I do not know whether this protein is made of a few subunits or not. So I can only write what I am quite sure. All functional proteins are tertiary. They're all globular. They function based on the shape. Okay. Idea of protein non-functional, frame shift mutation, or you say UAA is a stop codon. So that you get 1 mark. I think only one student mentioned that UAA is a stop codon. Only one student mentioned that. Okay, so which is very good. Right, so next question. So B, red blood cells originate from undifferentiated cells in the bone marrow that are capable of continuous mitotic cell division. State the name of this type of undifferentiated cell. It's actually multipotent stem cell, because this is a group of stem cells of the same family, which they will divide and differentiate into the family of blood cells. So it's got red blood cell, white blood cells. Under white blood cells, got lymphocytes, which are B cells and T cells, and also the B cells and T cells, lymphocytes. Yeah, B lymphocytes and T lymphocytes, and also the phagocytes, right, which include monocytes, neutrophil, eosinophil and basophil. They are all phagocytes, but you only learn neutrophil and monocytes as the phagocytes. Okay, multipotent stem cells. Answer is either you write stem cell or you write multipotent stem cells. Where's the answer? Here. Okay. Stem cell or multipotent. Stromal stem cell, non-embryonic stem cell, neutral cell, doesn't matter. Huh? Okay. Some students write pluripotent. Pluripotent is wrong. Huh? It's multipotent. Okay. So D is where it shows that some students have not mastered how to describe the graph. And some students just, I don't know, somehow rather ignore my advice. Because of that, you don't score full marks. Because I've advised many times, this is how you need to write your answer to score full marks for graph. Still don't want to write. Okay. Mainly because did not quote figures. A graph that's given, you want to describe, you have to quote the figures. So that's where most of the students lose the marks because they didn't quote the figures. How many percentage? I can't remember. Maybe about 1/3 of students or more than that, almost half, did not quote the figures. Even if they quote also, it's not complete. Okay, so you need to learn. So I'm going to use this opportunity for you to learn how to describe graph again for the last time. So when a graph is given for you to describe, first thing is check the overall pattern, the general pattern. So the general pattern here, you can see that there is a few parts to read. The first part is where there is, this is the first part where, you see here, the curve remain constant. So this is the first part. Second part is where it will increase all the way up to here, right? And the third part that it will decrease all the way up to here. So therefore, if you can see the pattern very clearly and divide the tree, it's actually very easy to score full marks, provided that you quote the figures, right? Some students didn't score full marks because they didn't quote the figures. So all you need to say is that it's very easy. You have to state the pair, quote the pair of figures. I cannot see. So I'm going to pull it up a bit so that you can see the value here. This is value in x-axis, is value in time in weeks. So I'll leave it this way. The very first thing you need to say is that from 0 to 2 weeks, isn't that easy? Then what is your y-axis is a mean hemoglobin concentration. So you just write there your mean hemoglobin concentration, okay, remains constant. So this is you describe. Then you quote the figures. Constant at where? You can put in bracket. Then you read off from the graph. This value is 12.6. So you write there 12.6. Some students didn't write the unit. A value without a unit doesn't make sense for science. So at least you must write gram per kilogram. You don't write body mass, it's okay, but you have to write at least up to here. So this one itself, you already get 1 mark. Quote figures means when the x-axis value is between 0 to 2, the y-axis value is 12.6, and the curve is constant. A lot of students didn't mention this. A lot, majority didn't quote this one. Okay, this is so easy to score. Number 2, then you say, okay, from week 2 to week 7. Sorry, week 2 to 7. So you say mean hemoglobin concentration, right? What happened to it? Increases, isn't it? Very straightforward. So increases, you tell me from what value to what value? Read from the graph. Of course, the original value already read is actually 12.6. No need to read already. So you read the peak value here, right? The peak value here is 15.2. So therefore, you just write there from 12.6 to 15.2, and then the unit gram per kilogram. This one you get another mark already. Isn't that easy? But a lot of students don't want to quote the figures. Just say, or increase and then decrease. How to give mark? Everything also increase or decrease, and then decrease or increase, and then constant. Where? Exactly where? Number 3, you see, systematically describe, you get all the marks. You know why we go round and round and round and round. Don't know what you write. I have to read until my eye also burst out already. 7 to 10 weeks, week 7 to 10. So you can say mean hemoglobin concentration drops. So drops from what to what? So from 15.2, you already read already, right? To, so the last value is right in the center. So to read from the center here. 13.7. So therefore this is 13.7. And then the unit gram per kilogram body mass. Okay? So that you get the marks already. So why 4 marks? Because just now when I write the 3 sentences, I can get more than 4. I can get more than 3 marks because in the sentence, I describe the pattern and I quote the figures in the same sentence. I describe, already get marks, and I quote paired figures. You see that I always quote the paired figures from, so when X changes from X1 to X2, Y changes from Y1 to Y2. So this is called paired figures. So because of that, I only write 3 sentences, but I get the full marks. So this is how you want to achieve write minimum and score maximum. Don't write maximum, don't write maximum and score minimum. I also read until I want to pingsan. All my eyes come out already. Read the whole passage and then get 0 mark. Okay. Right. Let's look at the mark scheme. Mark scheme, you got what? Describe and explain. So describe the graph and after that explain why. Okay. So here first is they accept if you write HB for hemoglobin and you write hemoglobin concentration for mean hemoglobin concentration, accept the unit gram per kilogram. Okay, or gram per kilogram like this. For gram per kilogram for body mass. So that one got no mark, just say that they accept. So what is the mark? Can you see they divide it into the constant region, increase and then decrease and then explain. So D means description, E means explain. So hemoglobin concentration remain constant at 12.6 gram per kilogram of body mass for the first 2 weeks or up to the start of injections. So this is described. Explain why it remains constant is because based on the earlier passage, remember I said you need to refer to the passage, there is sufficient oxygen, so the body did not produce EPO, so it does not stimulate red blood cell formation. So the hemoglobin concentration remains the same. Okay, so because EPO will stimulate red blood cell formation in the bone marrow stem cells. So when the red blood cell is being formed, that means to say there will be an increase in hemoglobin. So this experiment measure the hemoglobin hemoglobin in terms of mass of hemoglobin. Okay. So next, describe the curve I already mentioned. Increase in hemoglobin concentration from week 2 to week 5. So this is called describe. Okay. And then quote the data. So week 2 to week 5, so the value changes from 12.6 to 15.3, right? So this is called quote value. Or you can say overall increase by 2.7 gram per kilogram. And then you said decrease from what value? 15.3 to 13.7 gram per kilogram, or overall decrease by how much? So this is called quote value. So describe and quote value. Now explain why is there an increase and why is there a decrease in the hemoglobin mass. Because EPO that's injected into the body stimulate red blood cell production. And because of that, there's more hemoglobin produced. So strictly speaking, if you say because of EPO, more hemoglobin produced, I cannot give you mark. Because you didn't link the fact that EPO actually increased production of red blood cell. And because of that, that's why hemoglobin mass increases, because red blood cell contain hemoglobin. So strictly speaking, if I mark you correct, it's supposed to be wrong. If you only say hemoglobin concentration increase, because you're not explaining the full story. Why hemoglobin increase? Because red blood cell is being produced. Okay, so that's why the passage before this is so important for you. Draw a flow chart, draw whatever chart to see more clearly, so you know the important points to answer. Then decrease, explain why there's a decrease. Of course, because decrease, let's check the range where the decrease occurs, okay? So final EPO injection is here, right? After EPO is being injected, there's still an increase followed by decrease. So that means to say that if there's a decrease, that means my EPO that's injected earlier on must have been degraded. So I already break down the EPO, and probably the red blood cells that produce are also being removed. Now, how are old and unwanted red blood cells removed from our body? Is actually by phagocytosis. It's being phagocytosed. Some students say apoptosis. Apoptosis is wrong. Apoptosis is actually, let me ask one of you. Who I didn't call yet. Najiha, what is apoptosis? Najiha, what is apoptosis? Najiha. I've got students who write apoptosis. What is apoptosis? They say red blood cell die because of apoptosis. Najiha, what is apoptosis? You learn this in cancer. Very good. Programmed cell death. Right? Programmed cell death means this cell itself will die by itself because internally they got programmed cell death. But red blood cell cannot be apoptosis because they don't die by itself. They die because they're being phagocytosed. So therefore, students who write apoptosis is wrong. You cannot simply switch and swap and use any biological word as you wish. You need to make sure that the meaning of the word is correct and you're used in the correct context, in the correct place. Don't simply use, oh, the cell dies, so it's apoptosis. Sorry, it's not. The cell can die because of many reasons. Okay, so you need to learn up the meaning of the word. What does it mean? So EPO is degraded already. That means any chemicals in our body will not keep in our body for a long time. You will remove them, usually by phagocytosis. Okay, so EPO might be removed by phagocytosis. And then red blood cell may be removed by phagocytosis. So therefore, the hemoglobin concentration will drop. So that explains why there's a drop there. Okay, there's one part to explain why is it EPO is already stopped injected, but the red blood cell, the hemoglobin level is still increasing because it takes time for red blood cell. So let's say, for example, the EPO is being injected here, right? It takes time for it to circulate in the body to activate stem cells and bone marrow, and stem cells will start to divide, differentiate, form red blood cell. So because of that, it takes time for red blood cell to be produced. So therefore, the hemoglobin concentration is still increasing, even though this is a final injection. Okay, it takes time for the entire process of circulation, cell division, cell enlargement, cell differentiation. So this is happening over here. So during that time, cells are still being produced. So therefore, the hemoglobin concentration still increase. Okay, so then you already explained this part when it's constant. This part, why is it increasing? This part, even though after you finish your final injection, is still increasing. And this part, why is it decreasing? So explain everything. So let's look at the mark scheme. Okay. Describe. Increase after injection stop already. Yeah. Hemoglobin concentration continue to increase for 1 week after injection has finished. That means the final one already finished, because there's a time delay for red blood cell to be produced. Okay, after being stimulated, it takes some time for them to be fully produced. Okay. So just now I mentioned about why there's a decrease, because red blood cell got short lifespan. This stands for ignore. Ignore those who write apoptosis. You said phagocytose, they die, I give you mark. They die by apoptosis, I don't give you mark. In the same sentence, you got something that's correct and something that's wrong in the same sentence. I will not credit mark for that sentence. That means the first part is correct, the second part is wrong. The whole thing must be wrong. This is how examiner will mark. Okay, some students even better. The first part of sentence, the second part of sentence contradict each other. So you ask me to choose how to give mark. One said, oh, this one can, the other one said this one cannot. How to give mark? Of course, wrong, right? You don't ask examiner to choose for you. It's not MCQ. Okay, you need to tell me what is correct. Cell signaling stopped already, so cells are no longer stimulated. So why is that decreased? Because EPO already degraded. Whatever red blood cell produced already got short lifespans being removed, and then the cell signaling already stopped. Okay, AVP. There's a steady increase of hemoglobin because there's a steady increase in number of cells by mitosis, cell enlargement, cell differentiation. All this takes time, so that's why keep on increasing. As more and more mitosis happen, more and more cell enlargement, more and more cell differentiation. This is contributory factor for increase may be accumulation or increase in concentration of EPO with injection. That means it says that as you continue to inject EPO, it remains in the body, the concentration of EPO becomes higher and higher. So therefore, more and more red blood cell will be produced. Therefore, more and more hemoglobin will be produced. So that explains why it increases. Okay? So these are all possible correct answers, all possible correct explanation. Okay? So next question is the lung. So let me make it smaller so you can see the structure, the lung. So this is a gross structure of human lung. So you need to describe the part of the lung. Sorry, the description is given. So you need to state the name of the part and write the letter. So what I find that I think some of you know the name, but you do not know where's the location. A lot of students, you may give name correct, but then your location is wrong. Okay, so let's look at it. The first one is supported by incomplete C-shaped rings of cartilage. So that means it's B, isn't it? Okay, sorry. Supported by incomplete C-shaped cartilage ring. Say, okay, what do you mean by incomplete ring? This is a complete ring, right? So this must be incomplete ring, right? And then somewhere they said C-shape. So that means it must be trachea, isn't it? So the name of the gaseous exchange system is the trachea and location is B. So this is trachea. Okay. Next, lined by ciliated epithelium and supported by blocks of cartilage. So that means blocks of cartilage means fragmented cartilage. Okay, fragmented cartilage. So that means it must be in the bronchus, isn't it? So write your answer bronchus or bronchi, and then the labeling for bronchus is either C or J. Let me look at the mark scheme. Okay, C or J or C and J. Right? So next one, it is lined by Squamous epithelium is only in alveolus. So it said alveolus or alveoli. Location of alveolus is in D, right? Only D can be alveolus, or M. M can be alveolus, M. So answer is actually M, not D. It's not D. Okay, not D. Answer is M. Okay. Answer is M. Right. Why is it not D and M? Maybe because D is the surface of the lung, the tissue, the wall of the lung, whereas this is internal structure inside the lung. So M is the answer. Okay. So last one, lined by ciliated epithelium, but not supported by cartilage. That means this must be the bronchiole, isn't it? So except bronchiole or bronchioles. So where is the bronchiole? So this is the bronchiole, right? It's still in tube form, right? All these are bronchiole. All these are bronchiole. So answer is H. Okay. Now next question. In human blood that comes, in humans, blood that becomes oxygenated in lungs reaches body tissues without coming into contact with blood that's deoxygenated. So you need to explain how the blood that becomes oxygenated in lungs is kept separated from blood that is oxygenated. Now, if I'm going to mark this question strictly, I think a lot of students have to minus marks already. Okay, strictly speaking, right? So if this is the lung, right? This is the lung. Okay, this is the lung. So therefore, you will have got pulmonary vein that come from the left side of the lung and right side of the lung and go back to the heart. Right, the heart got two parts. So this is the left atrium, right? Left ventricle, right atrium, and right ventricle. So pulmonary vein will carry oxygenated blood to the lung, so it will not mix with pulmonary artery that carries deoxygenated blood from the right side of the heart. So this is the first thing, how it's being separated, because oxygenated blood is carried by pulmonary vein and deoxygenated blood is carried by pulmonary artery. So this is the first thing you need to mention. Second thing, when it arrives to the heart, the oxygenated blood will only go to the left part of the heart, which is separated from the right part of the heart by the septum. So oxygenated blood will go into the left part of the heart, whereas deoxygenated blood actually goes to the right part of the heart, separated by septum. So that's one mark to talk about the lungs, the heart structure. So this is the second best answer. So therefore, this is 2 marks. Okay, these are the 2 marks that I'm looking for. So of course, if a student says systemic circulation and pulmonary circulation, strictly speaking, I will not give mark, because any student can just give the keyword systemic circulation, pulmonary circulation, without really explaining in detail how. So strictly speaking, I will not give mark unless you describe to me. So how is it by having this circulation, you separate oxygenated blood from deoxygenated blood? Like this one here, this is the best answer. This is the best answer I'm looking for. Okay, so let's look at the mark scheme. So mark scheme says only oxygenated blood passes through the left side of the heart, which I mentioned already, or oxygenated blood in the left side and deoxygenated blood in the right side. So it's talking about that. separated by septum is the second marking point. So you only talk about heart structure, you also get the 2 marks already. And then the oxygenated blood is flowing in pulmonary vein, whereas deoxygenated blood is flowing in pulmonary arteries. Number 4 is a question mark. Okay, it's not very clear answer. I will not give mark for number 4. Right, double circulation. So what has double circulation to do with the question that they ask? Double circulation, pulmonary and systemic circulation. To me, this is not a good answer. To me, this one should not be given a mark. There are better answers than this. So I will look for this answer. If I'm examiner, in the discussion, I will reject this answer because it's just very vague. Can't see clearly. Okay, so therefore I close one eye to give you mark. Strictly speaking, no mark. Okay. Question number 6. So this is the last question. It's already 1 o'clock. I think we should stop, right? Okay, we should stop. We will continue to discuss the answer in the next lesson. Okay. Oh no. So tomorrow lecture, what should I teach? You'll be joining another class or lecture, right? Or tomorrow got no class, sorry. Wednesday. Wednesday, I discuss in another class. So I will only see you on Thursday, you know. So during this time, please go through the MCQ. Go through the MCQ, make sure that you check the answers. Looks like I only got enough time to discuss all the papers for this week. Okay. All right, so that's all for today lah. All right, so you all take care. Go through your answers and check, see how to score full marks, okay? I really hope you are working very, very hard, again, doing your best, really. Okay, all right, bye everyone. Have a nice day.